Okay, I think we can start. Good afternoon and welcome everyone to the third edition of PDCLife's, the workshop on parallel and distributed computing for life science, organized by me, Laura Antonelli, from the National Research Council of Italy, and by Salvatore Cuomo from University of Naples Federico II. The workshop is oriented to explore the key role of HPC in solving problems related to different branches of the life science. Unfortunately, because of the coronavirus outbreak, we haven't been able to meet each other in the beautiful town of Warsaw. Anyway, we hope you could take benefit from your virtual participation to the workshop and to the Europark conference. First of all, we would like to thank the authors and all of you to join our workshop and share your experience. And we hope you'll find new ideas and collaboration for your future work. We have to remember some deadline about the conference. After the workshop, we will contact the authors by email in order to participate on a special issue on the journal Parallel and Processing Letters to propose an extended version of the papers presented to the workshop. Instead, the final camera ready of the proceeding for the for the lecture notes in computer science have to be sent by the authors themselves using EasyChair, no later than 25 of September 2020. For any question about the special issue on parallel processing letter, you can contact me to the email address on the slide. Now I think we can start, and it's a great pleasure for us to introduce the keynote speaker, Mrs. Elodie Laine from Sorbonne Université. Elodie is an associate professor and she works at the Laboratory of Computational and Quantitative Biology. The title of her talk is Protein Sequence Structure Dynamics Function Relationships, Efficient Tools for Mining Experimental and Simulated Data. Thanks, Elodie, to accept our invitation, and let me know if you are ready to present your talk.
Yes, I'm ready.
Okay.
Thank you very much. I will try to share the screen.
Okay, I have just interrupted my sharing.
Yes, thank you. Is it okay? Can you see?
Yes, for me it's okay.
Okay. Okay, so good afternoon, everyone. So first of all, I would like to thank the organizers for the very kind invitation. So indeed, I work in the Laboratory of Computational and Quantitative Biology. So it's part of the Institute of Biology at Sorbonne University. We are located in Paris, so our buildings are close to the riverbank. You can see them on the bottom left corner. So the lab is comprised of about 50 people, and we are an interdisciplinary laboratory. So half of the teams develop models and algorithms to describe biological systems, and the other half perform experiments with the yeast as the main model system. So I work in the analytical genomics team. We're interested in problems related to genomics in a large sense. So from the DNA sequences to the 3D structures of macromolecules. So given a protein, we would like to be able to identify the sites that play a role in its function, to estimate the impact of sequence variations at these sites, to identify cellular partners and estimate their binding affinities, and to describe the functional states and the functional isoforms of the protein. So for example, we can take the beta-lactamase from E. coli, which is a protein involved in antibiotic resistance. So we may look at the sequence of the protein. We or we may look at the structure, exploit the information of the arrangement of the atoms in 3D space, or we may use both information. So here, of course, when I show this, there is a level of complexity that is hidden. So we don't look just at the sequence of the query protein, but we look at many, many sequences that are related to it because they share some similarity with it. And we don't just look at static structures, but we look at different conformations of the protein. So we look at the motions of the protein in solution. So I'm going to give an overview of different projects and tools that we developed, and then I will focus on one specific subject. So typically we start from the sequence of a query protein, and we retrieve sequences that share some similarity with it, that are related to it. And we make the assumption that all these sequences are linked by evolutionary relationships that can be encoded in a tree. So we exploit the topology of the tree to extract conservation patterns, which we use, for example, to predict the effect of mutations. So here I show a matrix where you have numerical values that correspond to predicted effects for all the possible substitutions. So in the 20 amino acids for the entire protein sequence. So we have about 5,000 mutations here, and the darker the color, the more deleterious the predicted effect. So another type of application that we can have is the prediction of protein interfaces. So contiguous patches at the surface of protein that interact with other proteins or RNA or DNA. So we have developed a suite of tools which allow to predict these sites using conservation measures and also using physicochemical and geometrical properties of the proteins, of the structures. So typically we are dealing with between 100 and 1 million sequences for each protein of interest. So to be able to efficiently handle these sequences, what we do is that we do not reconstruct the full tree that links all the sequences, but we subsample the sequences. So we extract representative subsets. For each subset, we build a tree, and we compute for each residue conservation levels from each tree. And by averaging over many, many trees, we're able to get statistically significant results. Another thing we try to do is to reduce the number of parameters we have to infer as much as possible. So by doing this, we are able, for example, for the prediction of mutations, our tool GEM is orders of magnitude faster than the state of the art, and it displays performance that are similar or even better for viral proteins. So another type of information we're looking at is structural information. So for sequences, we have many, many sequences that are available in public databases because of the development of high throughput sequencing. But for structures, we still have comparatively few data. So instead, we rely on simulated data. So we generate conformational ensembles by running molecular dynamic simulations. So our strategy is to exploit relatively short simulations, typically of a few tens of nanoseconds. And this allows us to run simulations on a large number of systems. So what I show here, for example, is a complex between PDZ domain and peptide, ligand peptide. In that case, we simulated the wild type and also 200 mutants, and we generated a matrix similar to the one I showed before. So here, because the calculations are much more demanding, we are limited in the size of the matrix that we can generate, but still we can do large-scale experiments. So we have developed algorithms that automatically extract biologically meaningful information from the MD simulations without requiring any expert knowledge on the system. So we have a framework based on graph theory where we compute the cross correlations between residues, and we define pathways in the protein structure that correspond to chains of residues that are correlated, move together, essentially. And what we observed is that we have a higher concentration of pathways, more pathways, longer pathways when we are in presence of a deleterious mutation. So this is what you can see here. You have two snapshots with the path that are in dark, in black, and you can see on top the mutant, the wild type, sorry, and at the bottom a deleterious mutant. Another nice thing here is that we can just, by simply exploiting the simulation of the wild type, again, using these pathways, we're able to identify the hotspots, so meaning the residues that are highly sensitive to any mutations. So these are the residues you can see in color on the right. And so compared to sequence-based methods, these methods are more computationally demanding, but on the other end, we have a vision and understanding of the mechanisms that can explain the mutational effects. So here we can see what is the role of the residues in the structural stability of the protein. And what we observe is that these hotspots generally form bridges between the ligand and the protein, for example, or between parts that are not very connected. Things like this. So another example is the identification of cellular partners. So here, what we generate are putative structures of protein complexes. So the team has been a pioneer in the realization of complete cross-docking simulations. So you start from a set of a few tens or a few hundreds or a few thousands protein, and you dock each protein against all the other proteins, including itself. So each docking simulation is relatively fast. It generates billions of conformations. So we typically exploit between thousands and hundreds of thousands of conformations. So because it's relatively, each simulation is fast and they don't talk to each other here, we can just run many simulations in parallel. And from this, we analyze the conformations based on different criteria. We also use conservation here, and we reconstruct matrices. So here you have all the proteins versus all the proteins, and the darker the blue color, the higher the interaction we predict, interaction strength predicted between the two partners. So you can see this as each protein has a number of potential partners, and you can kind of place them on concentric layers. So the further you go from the central protein, and the lower the affinity, if you wish, and you may have several proteins that bind to the same site or bind to different sites. So what is interesting in these studies is that we observed that it's important to know the global social behavior of protein to be able to identify its partners. So meaning that if a protein is sociable, it likes to bind to everybody, or if it's very shy and doesn't bind to a lot of other proteins. This information is important if you want to really identify its true partners. So that's the protein sociability, if you wish. Another interesting thing which you can see a bit in the matrix is that you have some squares. They're mostly white. So here the squares are defined by function. So you have functional classes of proteins that tend to avoid each other. Like here, for example, the enzymes, they really don't like to bind to each other. Whereas enzyme and inhibitors like to bind very much to each other. So you have a lot of predicted cross interactions here. Okay, so this is just something I already said. So if I want to summarize what I showed right now, these are examples where we exploit sequence data that are generated by high throughput sequencing technologies, and also structured data generated by high performance computing. So using molecular dynamics or docking engines, we develop abstract concepts and representations and algorithms to automatically extract biologically meaningful information. So we really try to be as automated as possible without requiring expert knowledge to generate large-scale predictions of different protein properties, so functional sites, mutational outcomes, cellular partners, and we can confront these predictions to experimental measurements that were obtained from high throughput technologies. So for example, for the mutational effects, we have access to matrices that are similar to those we predict, which are generated by deep mutational scans. So for example, the best we can do is for the beta-lactamase, a Spearman rank correlation coefficient of 0.783. So now I want to move to another subject, which is alternative splicing. So this is a regulatory mechanisms in eukaryotes by which multiple proteins are produced from the same gene. So you may think of a gene as a chunk of DNA sequence, but in fact it has a structure, so it's composed of exons and introns. And during the transcription, only the exons are going to be retained and are going to form the messenger RNA transcripts. And you may have different composition of different collections of these exons, so different versions of the transcript, which in turn will be translated into different versions of the protein, and these different versions may adopt different structures and perform different functions. So if you want to we can make an analogy with the trains in India, just to give you an idea of the type of events we can have, the type of variation we can have in the composition of the exons. So if we take a train from on this train line, we may start from Mumbai or Pune. So we have two different starts. We may go through Bhopal or Jabalpur, but not through both using the same train. So we have stations that are mutually exclusive. And we may take a little detour through Varanasi, so we may have an insertion with travel that's a bit longer. So these are typically the events that we see with alternative splicing. And this is for one gene. So if we have many genes, we may end up with a network that is quite complex. So in the last decades, we have learned a lot about the mechanisms that are at the origin of alternative splicing at the genomic level, and we have also learned a lot about the regulation of alternative splicing in different tissues. But there are still very few functional studies on the isoforms, so we don't, there are many still open questions that are unanswered and that cannot really be addressed with the current technologies. So for example, we don't know how many of the detected mRNA transcripts are really translated as proteins and functional in the cell. Another question is, how do new isoforms emerge and evolve? And what are the molecular determinants of the functional changes that are induced by alternative splicing? So we try to address these questions. So this subject is really an example where you have a variety of biological objects and data types that you have to deal with. So you really go from the sequence to the structure. So for example, we typically start with gene annotations that are available in Ensemble. So these are annotated transcripts for different species. So when you have this list of transcripts, then you may ask yourself, are these transcripts supported by RNA-seq data? Are they tissue regulated? And is it everything there is, or can we detect more? And to answer those questions, you have to look at the raw RNA-seq data. So for example, you can go to the BG database and you will have access to splice junctions that are based on RNA-seq data. And you will know the support you have, you will find more events, and then you may wonder, okay, now what is the composition of my isoform? Do they have particular domains? Can I have a domain that is completely truncated or missing because of the alternative splicing variations? So to answer this, you need to look at the PFAM database, where you have domain families that are defined with multiple sequence alignments that are associated, so you can easily map your transcript on this database. And from the domain composition, then you may go a step further and ask yourself about the structure of your isoforms. So what is the shape? What is the role of the regions that are alternatively spliced? what is the impact of the splicing on the protein structure and interactions. And for this, you are going to look at the protein data bank. So you really range from the sequence to the structure and the dynamics. So what we're doing is trying to develop tools that can integrate these type of data and analyze large-scale annotations with high efficiency. So we have developed a couple of tools, Thorax and Philosoph, to identify orthologous regions between across species, and to reconstruct transcript phylogenies and predict the 3D structures of the isoforms. So the goal of Thorax is to identify a mapping between different species, mapping of exonic regions. So it's not a trivial task because, so if you look at just genomic exons, you may have, for example, an exon in nematode that corresponds to 3 exons in humans. So you have to determine which part correspond to which part in the 2 species. So what Thorax does is it retrieves the annotations from ensemble, it cleans, it reduces the redundancies in the data, and then it combines pairwise alignments to cluster the exons that share some similarity with multiple sequence alignments that are applied on each cluster. And the multiple sequence alignments are constrained by the genomic coordinate of the exons. So this way, we're able to disentangle some, to remove some ambiguity that we may have because of high similarity between sequences. So our final goal is to define minimum building blocks for the input transcript that we have. So we introduced the notion of spliced exon or S exon, which is either a genomic exon or something smaller. And this is really the minimum unit that we need to reconstruct all the transcripts we have as inputs in different species. So Thorax reconstruct evolutionary splice graphs. So here is an example. So usually you have splice graphs that correspond to 1 species, but here we have the information of many species. And a splice exon is a node in the graph, and the color corresponds to the conservation. So the number of species where the node, where the S exon is present. So formally, an S exon is just a multiple sequence alignment where each line corresponds to a species. So for example, here I show you on top the visualization of the clusters that we have from the first step, where you can see that we have these 2 S exons, the one in blue and the other in purple, that are together. And then they're separated by the multiple sequence alignments. So here they're involved in a mutually exclusive event. Either you go from 3, you go through 3-0 or 3-1, but not through both. So you can see here that we are able to disentangle the orthology relationships across species from the paralogy relationships within a given species. And we may enrich our splice graphs by using the RNA-seq splice junctions. So here is a case study. This is the tropomyosin. It's a protein that is involved in the formation of actin filaments. So this is the splice graph computed by Thorax across 12 species, ranging from human to nematode. So we look at 1-to-1 orthologs of the gene. So maybe here we don't have in nematode, so maybe it goes up to zebrafish. So you can see that the graph is quite complex. Again, the color corresponds to the conservation. So basically the nodes there in yellow are just present in 1 species. So they have just 1 sequence, they're species specific. So if we ignore them, you can see that you have some patterns that are clearly identifiable. So here we have mainly events at the N-terminal, close to the start node, where we can have replacements of 3 S exons by another one. We have an event in the middle, a mutually exclusive pair, and also some events at the C-terminal part, close to the stop. So now we can look at the structure of this protein. So it forms a helix that dimerize to form a coiled coil. So here you have 4 copies of the protein in dark, and it is bound to actin to form the filaments. And in color, you have the exons that are involved in the events you can see on the splice graph. So now if we look at the RNA-seq data, I just show a zoom of the events in the middle, the mutually exclusive pair. So what we can see in the RNA-seq splice junction, again, here we have also 12 species. So we take the data from the BG database, which collect data from 37 experiments, and we have a tissue ontology associated to them. So you can see that we have support for these events. And we also have new events that are detected in the splice junctions and that are not present in the annotations. So you can go from 15.0 to 16.0, and meaning you don't have any of the mutual of the 2 S exon on the left and on the right. Or you can have both. You go from 15.0, 13.0, 7.0, and 16.0. So now you can look at the tissue regulation of these, tissue expression of these paths. So on the right, I show the person splicing values for the left and right path. Left is including 7.0 and right includes 13.0. And you can see that their expression patterns is very different. So first of all, there is the right path is more has more support than the left path overall, and you also have the right path that is present in hearts, which is really not the case for the left path. So another example is given by myosin 1b, which is also involved in the formation of actin filaments and muscle construction. So here it's a long protein, you have many, many S exons, but in the end, you can just see one event basically, which is the insertion of 1 or 2 S exons, 11.1 and 11.3. So again, looking at the protein structure, you can see that these S exons are in interaction with a partner, which is calmodulin. So you have 4 copies of calmodulin here. They're bound to these S exons, 3 S exons. And these S exons contains IQ motifs, which are motifs known to bind calmodulin. So on the right, I show the a multiple sequence alignment of the motif. So here, every line actually is a consensus sequence that represents an S exon. So it represents a whole multiple sequence alignment across different species. So what's interesting here is that these IQ motifs, these binding sites have been shown to have lower binding affinity for calmodulin compared to the other motifs that are constitutively spliced in the protein. And you can see that they lack in the canonical motif. We have IQ and also RG, and so it's on top of the alignment. So they lack the Q, and they also lack the R. So that could maybe explain why they have lower affinity. So you can go here again from sequence the splice graph to the structure and alignment. So we assessed the method thorax on a benchmark set of 50 human genes that we collected from the literature. They belong to 16 families, and we chose them because there was experimental characterization of different isoforms, and there was evidence for different functions of the different isoforms. So we analyzed them across 12 species. That's about 900 transcripts, and we had between 8 and 91 S exons. So we detected about 70 evolutionary conserved alternate splicing events, and about 2/3 of them were described in the literature. And we looked at the structural information we had for the S exons that were involved in these events. So we had structural information for a bit less than a half of these S exons. So this is in line with what previous studies that show that alternatively spliced regions are generally tend to be disordered. So we don't have so much structural information. We also looked at the function of these events. So for about half of them, we could find in the literature evidence that these events were involved in interactions. So this is also in line with what we can find in the literature, that there are experiments showing that alternative splicing rewires protein interaction networks. Okay, and by looking at the splice junctions from the RNA-seq data, we could increase the number of events by 35%. So that's a lot, meaning that we really have annotations that are partial, and we really need to look at raw RNA-seq data to have a more complete picture. So the other tool that we develop is concerned the reconstruction of transcript phylogenies. So when we have the description of the of the transcript based on the S exons, then we can think of really reconstructing evolutionary scenarios that can explain these observed transcripts. So we start from the observed transcripts, their description from thorax, and from the gene tree that we download from Ensemble. And our goal is to embed a forest, a phylogenetic forest, in that gene tree, where each tree in the forest corresponds to a transcript. So this is what we have on the right. We have three main evolutionary events. The creation of a tree, the root of a tree corresponds to the birth, the emergence of a transcript in evolution. We can have mutations along the branches, which correspond to gain or loss of S exon. And we can have loss of transcript in evolution, which will be a death, a dead end in the tree. So we start from the observed transcript in different species, and what you see here is just the description of each transcript with the S exon. So each cell has a different corollary and correspond to an S exon because we have this mapping across species. And when it's white, it means it's just not present in the transcript. The first step is to reconstruct the ancestral exon states at the genomic level. So we determine whether they are absent, constitutively present, or alternatively present. And then we sample the forest structures. So a structure for a forest is defined by the number of nodes, of subnodes that we have at each internal node, and the number of children for each subnode. And this basically fixes the number of death and the number of birth that we have. So we can just compute a lower bound for each structure and efficiently prune the structures that are not valid because they already have a too high score based on just on the death and the birth. So here our algorithm relies on a multi-start iterative strategy. So basically we do a random jumps in the space, and then when we find a valid solution, we explore systematically all the solutions around it. So because we do these random jumps, this is very easy to parallelize. You can just run different simulations and take the best you have over these simulations. And when we find a valid structure, then we identify the phylogeny, the best phylogeny associated to it. So the best transcript assignment, which transcript are going to be paired up in the tree. So we have a score which is computed using the maximum parsimony principle. So the best phylogeny is going to be the most parsimonious one, the one with the lowest number of evolutionary events. So we use Sankoff algorithm. So here is an example of results that we obtained for the C-gene N-terminal kinase family. We have here 3 genes, gene K1, 2, 3, 7 species from human to nematodes, and 60 transcripts, which are the leaves of our forest, and they are linked by 7 trees. So each tree is colored differently. And what we did here is we chose not to penalize the death of a transcript because of the under-annotation problem that we saw previously. So we know that there's some species that are under-annotated. So by not penalizing death, we are able to find, to make the phylogenies go upper, let's say. So what we do also is we predict the 3D models of the protein isoforms. So now we can also look at the 3D properties of these isoforms. So an interesting thing is, for example, the transcript that you see on the leaves, which are gray, they don't have any parents, so they don't have any tree, any phylogeny associated to them. So we call them orphan transcripts. So now if we look at their properties, we can see that they're smaller, they have bad stereochemical properties compared to the other ones, and they are more exposed than the parented transcripts. So here in the box plot, the orphans are on the right, it's the dark gray, and the other ones are light. So you can see the distribution is wider, and they have these properties that are really different from the parented transcripts. So that's a good thing, because these are unlikely to be functional because they are not conserved in evolution, so they also show peculiar properties. Then what we did with this tree was to date the appearance of a well-documented alternative splicing event, which involved 2 pairs of mutually exclusive exons. So at the C-terminal end, you can see a variation, and also in the middle of the protein. So we dated this in the ancestor common to mammals, amphibians, and fishes. And we ran molecular dynamic simulations to elucidate the molecular determinants of the functional role of this variation. So it's been shown that the 2 isoforms have, so for the C-terminal part, we don't have any information, but for the part in the middle, the variation induces different selectivities to protein substrates. So what we could see in the simulations is that we have side chains that are much bigger in one exon compared to the others. And this is associated to a larger flexibility, exposition to the solvent. So we could here highlight some key residues that may be responsible for the different selectivities to the partners. So this is also a region that we predict as interacting with partners. Another interesting thing was the discovery of a new isoform that is not documented in the literature, which is shown here in pink, and it's quite conserved. As you can see, again, it emerges in the ancestor common to mammals, fishes, and amphibians. So we simulated this one as well, and so it displays a large truncation. It doesn't contain any of the previous the exons I showed previously. And this implies, or this induces a large motion of the activation loop of the protein. But in the simulation, we could see that the protein remains stable. So the catalytic site is a bit altered, so it may not be catalytically competent, but it may have a role in the cell, for example, for dimerization. As we saw, it was stable. So the perspectives now for this work is to analyze the whole, to apply these tools to the whole human genome. So for example, we ran Thorax on all the 20,000 protein coding genes, human protein coding genes. It took 19 hours and 15 cores, so it is quite efficient. We have for about half of the genes, we have one-to-one orthologs up to zebrafish. So this is quite substantial amount of data that we can really analyze. We have between 30 and 350 S exons per genes, and up to 6,000 residues within one S exon. So in the future, what we would like to do is to, so we want to create a knowledge base based on these results, and we would like to scale up to the analysis of transcriptions of human individuals from different populations. So these are data that are coming in the next years and that will require a lot of computing power and analysis power. So this is also why we try to develop efficient tools. And we would like also to try to develop deep generative models that would just learn from the patterns, the events that we detect as evolutionary conserved, which are likely to be functional, and which would try to generate some alternatively spliced, alternate splicing inspired variations on protein. And we could apply this also to bacterial proteins, for example. Okay, so with this, I want to thank you for your attention. I want also to thank people from the lab and collaborators. So in particular, Alessandra Carbone, who is the head of the team and of the lab. and Hugues Richard, who is in charge of the alternative splicing project with me. And I'll be happy to take any question.
Okay, thank you very much, Elodie, for your interesting talks. Are there any question on Slack? No, no. All participants can do any questions on Slack if they want. I have just a curiosity about your simulation. What is the speed up gained in your simulation? For example, what is the computational time without without use of any parallel tools?
So this would be a bit crazy, I think. So if I go back to the first examples. So here, typically for the MD simulations, what we do is we run the simulations on 16 cores. So these are not really, they don't really, these are simulations that if you don't have a special purpose hardware, you don't really scale up very well when you have nodes that need to communicate. So we tend to stick just on one node. So it's about maybe 1 week to have one simulation. And then because we have relatively short simulations, so there are really two strategies here. You have people who will run very long simulations, and then they will observe many events. So our strategy here is really to have just some sampling around the initial state.
Oh.
So we run several replicas, of course, for each system. But then, because we have short simulation, we can really benefit from the from having many cores.
Okay.
And here, docking is like nowadays you can have a docking in I don't know, less than 1 minute maybe. So, but then if you don't parallelize for the complete cross docking, it's gonna take ages.
There is a question on Slack. If you prefer, I can read the question for you, or you can.
Okay. Because if I want to read, I don't know. Do you still see
okay.
my?
Is a curiosity about the importance of the loop codes and parallel tools in your work, and the programming language. What other programming language used? What I think is this the question? What is the middleware used? The parallel architecture tools and so on used in your simulation?
So here, well, when we use engines that we don't develop, so for example, for the docking and the MD simulations, we don't develop the code. So then the programming language can be anything. So here MD simulations is typically Fortran, I think. And for the cross-docking, it's probably C, C++. Then for the tools that we develop in the splicing business, we have Python for Philosoph, and Thorax is developed in Python and Julia. So we parallelize with Julia. And for the architecture, well, we are not, so far we haven't run anything on GPUs, but probably we should for the MD simulations at least. Otherwise, it's we would just run it on the we don't require that many RAMs, so I mean, we don't have really special requirements other than a regular cluster.
Okay, thank you again, Elodie, for accept our invitation, and the discussion can be continued on the Slack, all the participants, okay? And we can pass to the next talk. by, thank you, yes.
The problem is I don't see my mouse anymore. That's a bit of a problem. How am I going to do?
Okay, thank you for stopping your sharing. And the next speaker is Mr. Luca Fiorillo from University of Naples Federico II. The title of his talk is Analysis of Genome Architecture Mapping Data with the Machine Learning and Polymer-Physics-based tool. Luca, let me know if you are ready for the talk.
Yeah, hello. I don't know. I think it will be streamed on. Exactly.
Okay.
Yeah. Okay.
Okay.
I'm
Thanks.
ready. Okay. Thank you.
Hello, I'm Luca Fiorillo from the University of Naples Federico II, and I will talk to you about analysis of genome architecture mapping data with a machine learning and polymer physics tool. First, a little bit of background. We know that the three-dimensional DNA organization is linked with its functionality. For instance, a gene, in order to work properly, must be brought in contact with another region of DNA composed of gene activators. So DNA must shape itself in a manner that each active gene is especially close to its own activators to ensure the proper functioning of the cell. That's why there's been a huge effort to investigate the three-dimensional organization of DNA in cell nuclei. And only recently there has been a real experimental breakthrough, as technologies capable to investigate the DNA 3D structure genome-wide were invented. Now, the output of these technologies is quite similar, and it is sketched here. Given a DNA region virtually split into equal portions called bins, the output is a matrix whose entries, say the entry 1, 2, circled here in red, contains the contact frequency between bins 1 and 2. That is to say, it contains the frequency whereby those two bins were found contacting each other across a whole population of cell nuclei, which in turn is a proxy for spatial proximity. Now, two important examples of this kind of technologies are IC, or Genome Architecture Mapping, shortly said GAM. And I show you here an example of GAM output data, just to point it out that the contact matrix is typically represented by an heat map, where in this case whites stand for low frequency values and reds for high frequency values. Here on the right, I'm just pointing it out that since the matrix is symmetric, in literature sometimes you see reported only the upper triangular part of the matrix in this manner. Well, with this great experimental advancement, of course, a huge amount of data were produced and now are required to be understood. So, to make sense of the data collected by these new technologies, polymer physics models and computational tools are being used. In the next slide, I will shortly review the Prisma computational tool, which was designed exactly to tackle this kind of problem. Indeed, the Prisma method is a computational tool designed to derive the possible 3D structures of a DNA region, starting from experimental data. Clearly, we want these structures to be physically meaningful. That's why Prisma must be informed with a polymer physics model, in our case, the SBS model. This model represents a DNA filament as a self-avoiding polymer chain composed of beads, surrounded by diffusive molecules called binders. Now, bead and binders have a specificity of interaction, represented here by a color, meaning that only same-colored beads and binders can attract each other. And binders, by attracting simultaneously more than one beads, drive the folding of the polymer. Now, I won't go any further into details. I will just mention that the SBS model was proven successful in reproducing many experimental findings about DNA. So now let's take a look to the Prismar pipeline for a given DNA region. We give as input for Prismar an experimental contact data for that given DNA region. Then we also use as input a random SBS polymer with a random number of colors and distribution of colors along the chain. The first goal of Prismar is to find the SBS polymer best reproducing the experimental contact data. Or, in other words, we want to train the SBS model over the experimental contact data in order to get the best SBS polymer compatible with such data. We do this by implementing a learning procedure which gets us the SBS polymer trained on the initial data. Once we have this polymer, we produce hundreds of replicates of it. We put each replicate into an initial random configuration in space inside a simulation box, and we perform for each replicate molecular dynamics simulation until each polymer reaches thermodynamics equilibrium. This way we end up with an ensemble of three-dimensional structures which are supposed to be representative of the real three-dimensional configurations that the DNA region under study can assume inside nuclei. Clearly, the molecular dynamics simulation step is the most challenging from the computational point of view. That's why they require HPC resources. And to give an idea of the resources involved, for each run we can employ from 8 to 64 processors with computational time ranging from 24 to 48 hours according to the polymer size. Well, this is an overview of Prismar, and so the next question is, how does Prismar perform on GAM data? I'll answer this question by showing the application of Prismar on GAM data of a 6 megabase DNA region named the SOX9 locus, a portion of mouse chromosome 11. To show you the effectiveness of Prismar on GAM data, I will first describe a consistency test. That is to say, we start from the experimental GAM data of the SOX9 locus, given as the upper part of the contact matrix, and then we apply Prismar on such data. As seen before, we end up with the ensemble of three-dimensional structures representative of the SOX9 locus, and then over this ensemble of structures we simulate a GAM experiment, meaning that we run an algorithm simulating the GAM protocol over such structures. We obtain in this way a simulated GAM matrix, which we can compare with the input experimental one. And you see that the comparison is very nice. Look also at the Pearson correlation coefficients between the two, allowing us to say that the Prismar method successfully reproduces the GAM input. But this is a consistency test, as said, because Prismar was designed exactly to do this, to reproduce the input data. So, as more stringent test, we do now the following. Over the ensemble of three-dimensional structures, we now perform a simulated high-C experiment. again running an algorithm simulating the IC protocol, which was developed in previous publications. We obtain in this way a simulated IC matrix, which we can compare with the experimental IC data of the SOX9 locus. That is to say, a completely independent dataset. Strikingly, the comparison between these two matrices is very good. Look again at the Pearson correlation coefficient. So by using three-dimensional structures that Prisma derived from GAM data, we are able to reproduce a completely different experiment. So we can say that Prisma structures can work as proxy for the real configurations of DNA in nuclei. So now, what can we do with these Prisma-inferred three-dimensional structures? Well, an important application is that we can find the preferred location of important DNA regions in the chromosomal architecture. To understand this, let's focus on the case of the 16p11.2 region on mouse chromosome 7. Mutations on this region are linked to autism. So understanding the DNA sites which are nearby it can be crucial for biomedicine. And so with Prisma, let's derive the ensemble of three-dimensional structures for chromosome 7, starting again from GAM data. This is an example of three-dimensional structure derived by Prisma for mouse chromosome 7. On the bottom you see the color scheme for this structure, and in particular just look at the red region containing the 16p region. In this example we can say that the 16p occupies a very peripheral position, but this is just for this single configuration. Let's do a more systematic study of the position of this region in the following way. Let's analyze the distribution of the positions of the 16p region across the whole ensemble of three-dimensional structures derived by Prisma. So to define the position of the 16p region, we use the so-called location ratio, that is to say the ratio between the distance of the region from the center of mass of the chromosome and the gyration radius of the whole chromosome 7. In this way, much bigger than one location ratios indicate peripheral positions, while location ratios close to 0 indicate internal positions. So, the blue histogram is the distribution of location ratios for the 16p region, while the gray one is the distribution of ratios for randomly chosen region over chromosome 7. You see that the two distributions are not compatible with each other, as shown by the Mann-Whitney compatibility test, and in particular the distribution of the 16p is more shifted toward higher values of the location ratios, and in particular we find that 10% of the times the location ratio of the 16p region is above 1.5, indicating that the 16p region occupies very peripheral positions. So we can say that this first analysis, exploratory analysis, indicates that the 16p can occupy peripheral positions. And this suggests that the 16p can form contacts with other chromosomes, which can be important for experimentalists to design experiments focused on interchromosomal contacts to understand, for instance, the arising of the autism disease linked to this region. So, conclusions and perspectives. Computational tools combined with polymer physics models are instrumental for the study of DNA 3D architecture in cell nuclei. The computational tool Prisma can infer the possible 3D structure of a DNA region from GAM data, and we have seen that for Prisma to work, its PC resources are essential. Finally, deriving the possible 3D structures of DNA regions can provide important insights on the contact pattern of genes linked with diseases. This could help designing focused experiments. Thanks for your attention.
Thank you, Luca, for your interesting talk. And let me see if there are any questions on the Slack. No, there aren't. So, Luca, I have a curiosity about a parallel architecture you used in the test. Did you use the MIMD architecture or multi-core?
Yeah, it's a multi-core architecture, and yeah, it's also, we use the parallel software we use for molecular dynamics is quite standard. It's the LAMMPS language, and so it's already designed to work on multi-core architectures to optimize the running. Yeah.
Okay.
Yeah. But basically, it just decomposes the space for the molecular dynamics simulation, and so they just then passes the results of the question each other.
Yes, and without use of any parallel tools, what is the computational time of the entire simulation of your?
Without parallel.
Yes. Is
yeah.
it possible or isn't?
I think it will be, I don't know, a challenge for the patients because.
Okay.
Because I think that right now when we run
At least with the 6 cores. Yes, I have understood.
Yeah, exactly. If we
Okay.
use with minimum 6 cores, we can have runs that last a full day, 24 hours, according to the polymer sites, but more or less that is the order. If we had to use just one processor, so without parallelizing, actually, I cannot imagine. Maybe it could be something of weeks.
Okay. Thank you.
Thank you.
Thank you again, Luca. Are there any questions, curiosity about this work? Okay. I think that applicants, participants can view your videos on YouTube, and they can do any questions. So also in the future day.
Yeah.
Okay. Thanks again, Luca. Now we have a coffee break till 3:30. And we can continue with the workshop after the coffee break. See you later to everyone.
Okay, see you later.
See you later.
Bye, see you later.